And then in the front part is the aqueous humour that fills this anterior chamber here, and also the posterior chamber, which is the thin layer between the lens and the iris here. And the aqueous humour, mechanically, it's pretty much like water in its consistency, and it flows because it is supplying nutrients to tissues and removing waste products. So it's fulfilling the role that blood fulfills in most organs, but in the eye you need a transparent fluid, so we can't have blood, we have aqueous humour instead. So to zoom in further details, very beautiful picture drawn by Masha. The aqueous humour is produced at these ciliary processes, which are these sort of frond-like things here, roughly a constant rate. It flows radially inwards. You have to rotate this and imagine it's in 3 dimensions. So it's flowing radially inwards between a very narrow gap between the lens and the iris here. Hi, Andrew. So it flows through this gap. It then flows anteriorly, so forwards towards the front of the eye. This is here, I should say, is the part that you can see, the part at the front of the eye. So it's flowing forwards through the pupil, which is just a hole. And then in the anterior chamber, it's a very complicated flow actually, but the net flow is radially outwards, and the flow goes towards this angle here of the eye between the iris and the cornea, the front part of the eye. And then it flows through the trabecular meshwork, which is a loosely packed tissue with lots of gaps through which the fluid can flow. So it's got quite low resistance. So most of the fluid goes through this path to Chernyavsky canal, which is, if you rotate this in your head, it's a choroidal vessel, and the Chernyavsky canal drains into the venous network. Okay, so that was what was originally known about until about 50 years ago, and then another pathway was discovered whereby there's a fairly low resistance path down here, so through the back of the base of the iris root, and then the base of the ciliary body, and then down into this choroid.
Sorry, just trying to make this a bit
more accessible.
Better see and
is that stable?
Yeah, I think it is stable.
Okay, because I'll just go back and show you the choroid. So this is the vitreous humour is surrounded by the retina, which goes around the back of the eye, which is obviously collects the light signals and turns them into electric signals that the brain can interpret. And then outside of that is a very dense layer of blood vessels, which is called the choroid. And it's a dense network of blood vessels that supplies the base of the back of the retina. And then outside of that is the sclera, the white part of the eye that you can actually see if you look at somebody's eye. So what was I saying? Right, so this flow goes down through this choroidal tissue. So the choroid, as I said, is full of blood vessels, but this flow is going in between the cells. So it's not going down blood vessels, it's going in the tissue of the choroid, and travels backwards. But meanwhile, as I said, the eye is pressurized, it's at quite a high pressure. So also there is a seepage through the sclera into the tissue outside. And this is the unconventional, so the original drainage that I showed you into Chernyavsky canal, that's called the conventional drainage. And then this one is commonly termed the unconventional drainage, going down into the choroid. And or sometimes uveo-scleral flow, because these tissues here are called the uvea, and then the sclera part. So it's either, I use either terminology. They're not quite the same, but roughly for the purposes of this talk, we can assume they're the same. Okay, so this was the original sort of seminal paper in which this flow was discovered in 1965, and it was in experiments on monkeys, sadly. But this guy, Anders Bell, found about a third, at the normal pressure of the monkey eye, about a third of the fluid was draining via this unconventional route, and 2/3 was draining via the conventional route. And then he also raised the pressure to double, so it's 11 millimeters of mercury in a monkey eye, slightly higher in a human eye, and then he raised it to 22 millimeters of mercury and found the rates then. And if we plot those on a graph, the conventional flow is shown here. So at the higher pressure, you can see there's a much higher flow. It increases by a factor of almost 5. And it's generally accepted that the conventional flow is linearly dependent on the pressure, because the resistance to the outflow is constant at different pressures. The unconventional flow has not been well studied, so we don't really know what's happening. But the interesting thing from this paper is if you raise the pressure in the eye, it seems like the unconventional flow hardly changes. It does change by a factor of 50%, but it's not a big increase. There are lines that I can't see. Okay, so I did draw a few more lines which would have helped orientate you. But anyway, it's only a 50% increase. Okay, and then later, the same person, Anders Bell, with a collaborator, Phillips, found this flow, or demonstrated the existence of this flow in humans. So patient was injected with radioactive isotope of iodine into their anterior chamber. So this is just a cross section. This is just a photograph of a cross section of the eye, and the anterior chambers at the front is looking upwards in this picture. And then on the right is shown the radioactivity after a week or so, I think. And you can see that the anterior chamber, there's a lot of radioactivity there, as expected, because that's where it was originally injected. But then a small amount of radioactivity is seen coming round, which you can sort of imagine is the start of a circle. So it's demonstrating that this flow exists in humans as well as monkeys. And it seems to exist in all species. So there was this intense research done by Anders Bell in the 60s and 70s, which established this flow and various properties of it, and then research on it sort of died down and hasn't been looked at since, because it was thought to be not that interesting. Meanwhile, our collaborator, Alex Voss, who suggested the problem, thought that maybe this would be helpful in the clinic for this problem. So I'll just tell you about this. So this is something that's most commonly used to treat age-related macular degeneration. That's a very common condition of the eye and affects about 1 in 40 people over the age of 50. So you probably, especially if you have older friends, you will probably know somebody who has this condition. And if it's the wet form, which is also quite a common form, it is often treated via this intravitreal injection. Its intravitreal injections are used to treat all sorts of things, and here it's being shown to treat a leaking blood vessel. But I believe the most common reason is for age-related macular degeneration. Anyway, that doesn't really matter. But the point is, what happens is you inject the drug that is treating the condition into the middle of the vitreous gel, and so you get a blob of drug in here, and over the course of time, it moves around. And actually, in research I did a long time ago, we showed that primarily it moves around because of eye rotation. So as you move your eye to look at different things, you rotate the eye and you mix the drug in, and eventually some of it, a small fraction of it, will get to the point where it's needed and deliver the drug to the macula, the sort of most sensitive part of the retina. Anyway, that's all very interesting, but not for this talk. But the point is that Alex was wondering if maybe you could instead use the uveo-scleral flow. So if you just use eye drops, put it onto this top surface of the cornea, the patient can do that very easily. Then the drug will diffuse across the cornea and go into the anterior chamber. And then, just like I showed that radioactivity in the previous slide, some of it will be carried around the sides of the eye, and eventually, hopefully, some of it will end up, very small fraction, but some nonetheless might end up at the site where it's actually needed, at the back of the eye. And that, if you could use that method for getting these drugs to the back of the eye, it would be save a lot of clinical time, and it's obviously a lot less unpleasant for patients who don't really like having these things done very much. So that was the sort of the motivation. So a model. This left-hand diagram shows you roughly the anatomical arrangement of the tissues, and then on the right I've done a greatly simplified picture that just is slightly easier to put labels on. But what we have is the sclera, the white part of the outside of the eye, and then internal to that is the choroid, And then there is nothing particular adhering the choroid onto the sclera. So surgically, you can quite easily get in between those two layers and put things in there if necessary. And it's a bit controversial amongst different clinicians, but there could be a space in vivo between the sclera and the choroid, and really only a tiny space, but nonetheless, if there is a space, that would greatly affect fluid flows, because there's a gap, and fluid would obviously flow much more easily through a gap than just between the tiny gaps between cells. So potentially we can have what's called a potential space between the sclera and the choroid. And then on the other side of the choroid, so the inside of the choroid is going to be the back of the retina. It's called the retinal pigment epithelium. It's a membrane that controls fluid and solute transport across it. Okay, so in this right-hand diagram, I've made things simpler. I've just put theta on a horizontal axis. I have, and so fluids coming in, this is the uveo-scleral flow coming in from the right. If we look at the black arrows, that's water flow. The retinal pigment epithelium RP is pumping fluid across it. And then fluid, as I mentioned, it goes out across the sclera. We also think that solute transport might be important in this problem as well. So we also consider flow of albumin, which is one of the primary constituents of blood. Actually, before I said that, I should perhaps say for a start, the choroid, as I've mentioned, is very dense network of blood vessels, one per unit mass of tissue. It's one of the densest or the most volume of fluid going through per unit volume of tissue. So there's an awful lot of blood vessels in there, and those blood vessels will be leaking, especially at the capillary level. They're very leaky, and choroid is very highly fenestrated. It has a lot of holes in it so as to aid nutrient transport and so on. Okay, so also within the choroid, what's happening is fluid is leaking primarily out, well, definitely out of the vessels because they're at a slightly higher pressure than the surrounding tissue. So you get contributions of fluid within this tissue. And also, as I was getting ahead of myself, but you also get potentially albumin coming out of the vessels as well, because there's a lot of albumin in blood, and that flow of albumin might also affect things. So we use albumin as a kind of tracer to mark any solute that's in the blood, but it is one of the most the most important. So yes, so we have no albumin crossing the RPE, because that's controlled. We have albumin coming out from the choroidal vessels. We might have albumin coming in through the entrance, and then albumin also crossing the sclera as well. So I think I've probably said most of the things on this, but so as I say, RPE is prescribed, so we're going to set that in our model, the flow of fluid. The choroid, we have the transfer of fluid and albumin between the tissue and the capillaries, and we're going to split, just for reasons of convenience really, split the capillaries into two types. Red ones, which are arteriolar, sort of the arterial end of the capillaries, and then the venular end of the capillaries as well. So most of the transfer is happening in either capillaries or very, very small vessels. But mathematically, actually, this splitting doesn't really matter because they all just get lumped into one term in the equations. Then we have, the choroid's going to be modelled as a porous medium, so because we're thinking about flow between gaps in cells. Potential space, well, we don't actually know what's in there. It's not even known, as I say, whether it exists. So we could do a porous medium flow if there's stuff in there, or we could just consider it's open. I mean, it could be a tiny gap, just a micron or even less in width, and it would still be very significant. So we could do Stokes flow, which would be just an open space there, modeling an open space. And the height of this potential space is going to depend on the pressure. So the greater the pressure in the in the gap, the wider the width of the gap. So I've shown that with sort of these springs here. I'm modeling that. And then on the outside is the sclera, and that's also going to be modeled as a porous medium effectively, but with a much lower permeability. And there's data for that in the literature. So I mentioned that the potential space was modeled using a sort of spring model, and this is the spring law that we use, so that the potential space can't collapse to a width below 0, so it has to be above this line. So really what we're doing is a linear spring law for large, larger gaps, and then it goes down towards 0 for small and negative pressures. And yeah, this is a law that does that, and we usually use n equals 1 simply because we can then invert, it's a quadratic equation, we can invert it, so it's convenient. Okay, so more details of the equations. I mentioned that we were doing, these are just the standard equations for porous medium flow, if you don't know them, and if you don't know fluid dynamics, but this is just standard. The only interesting thing really about this is we've got this flux out. So in the choroid, there's potentially fluid, well, there is fluid coming out from the vessels. So this is creation of mass effectively within this tissue. So QC here is a volume of fluid per unit volume of tissue per unit time. And it's the sum of the contributions from the two different types of vessels. And each of these is going to be proportional to pressure difference, which is mechanical pressure plus oncotic pressure. Okay. So that was the flow in the choroid that I was just talking about. Then we've got flow in the potential space. I'm just going to talk about the case of a gap because it's a bit more interesting, and it avoids us needing to find the permeability for this layer, which we don't know anything about it. So it's nicer in that sense. So Stokes equations, and these are completely standard fluid equations. Then boundary conditions. So we have 3 horizontal boundaries. So the bottom boundary next to the sclera, as I said, we model the sclera as a porous medium. So flow across the sclera is given by permeability of the sclera. This is again Darcy flow times the pressure drop divided by the thickness of the sclera. It's going to be HS, and then mu here is the shear viscosity of the fluid, which we're modeling as water, so we can get that that constant. At this interface, we have continuity conditions. We need, this is perhaps a bit of technical detail, but we do need the transverse, the velocity of the Stokes flow parallel, and we just set that to 0 at this point, but otherwise it's continuity conditions. So that's there. And then at the top, as I said, we have the prescribed inflow, which is set. Okay, so that was the horizontal boundaries. Then the vertical boundaries, so at the right-hand end, that's the inlet. The flow has already crossed, as I mentioned several slides ago, it's already crossed that, so it's come from the anterior chamber, it's crossed the iris root, and it's crossed the ciliary body, the ciliary muscle to get there. So we model those two pieces of tissue as having some kind of resistance, and then we have the pressure drop, so IOP minus the pressure here at the inlet, the IOP, sorry, intraocular pressure, which is the pressure in the anterior chamber, that pressure difference is equal to resistance times the volume flow, volume per unit time, Q. There. And then at the posterior panel, I should have said the whole model's axisymmetric. So this is on an axis of symmetry. So we have symmetry conditions there. Okay, so that was all the equations for the fluid. We also have to model albumin, and if we're thinking about, I mentioned we were putting a drug in potentially as well. So the solute equations go the same for either albumin or a drug. So I'm going to do them together, and I just call this thing X, and then it could be either A for albumin or D for drug. Okay, so we have this is J is the amount of solute per, so the moles of the solute per unit area per unit time, flux is called, is equal to, this term represents the solute being carried along by the flow, so velocity times concentration, and then there's a component due to diffusion, which is modeled by Fick's law, very standard way of modeling here. So this is the flux, and then we have divergence of the flux is equal to, now we have potentially mass creation. So this X, C is the amount of solute, moles per unit volume per unit time being created. So that is from these blood vessels, and we need an equation to get this. So that's equal to contribution from the red vessels plus contribution from blue vessels here. Okay, and I'm going to define that later. And then in the potential space, you have the same equations, but there's no mass creation. So we just have a 0 there. Then we have boundary conditions, bottom, I'm going to define that later, middle continuity conditions as before, and at the top, no flux by definition. And then at the inlet, we're going to say, so then it differs. For albumin, it's going to be well mixed. In the anterior chamber, we assume whatever's coming in is just well, already mixed. And then for the drug, though, it's coming in at a certain concentration, which is prescribed depending on the concentration in the anterior chamber and so on. That's a complicated calculation in itself, but it's given by a number. Okay, and then at the posterior pole, symmetry conditions again. Okay, I should keep an eye on the time. Okay, good. Right. Oh yes. Okay. So this is, I said that the flow across the sclera and also the flow out of the blood vessels, I said see later, and this is the see later bit. So we're going to model the flows across both these membranes, so across the walls of these little vessels here, and also across the sclera is modeled by these Chernyavsky equations, which are standard equations for modeling flow across membranes. So JV here is water flux, is the volume per unit time of water going across, which I've already mentioned. So that's equal to some constant times the mechanical plus osmotic pressure difference. And then JS, which is the solute flux, this is moles per unit time, is given by a constant times the change in concentration. So the albumin will flow and drug will flow down the concentration gradient, and that's expressed in this term here, and then plus a term which is flowing with the water flux multiplied by a factor that's to do with the ability of that molecule to get through the membrane. Okay, so we do that with different constants, obviously, but both for the sclera and for these walls of blood vessels. Now, as you might have noticed, this is a very long and thin domain, and the width of the choroid is about 50 times smaller than the length that the fluid's going to go through. So a very obvious thing to do is to use different scalings in both directions to simplify the equations, and that's very successfully used for many biological models. So length scales we choose. So we're going to non-dimensionalize. Length scales are based on these physical dimensions. Velocity scale, we know what the total rate of production of aqueous is. We know that this flow is maybe about 10% of that order. So we non-dimensionalize velocity scale based on 10% of the total volume flow. And then we use those to calculate pressure scale based on the Darcy equation that I showed you. Within the potential space, we use a different set of non-dimensionalizations. The length scales are based on the dimensions of that space. And remember, I mentioned that, so the choroid is about 200 microns thick. The potential space may only be 1 micron or, that kind of order. So it's much, much smaller. And so we're going to non-dimensionalize on different length scales. And pressure scale, we're going to use the same pressure scale from here, but then that allows us to work out the velocity scale from the Stokes equation. So we have a different velocity scale, and it's potentially quite different order of magnitude. We can then solve for the R dependence. If you've done this kind of simplification, you'll know how it goes. You can solve exactly for all the radial dependence in all variables. And it turns out that the pressures and concentrations very nicely are independent of R. So pressures, and by continuity, that means the pressure in the choroid, so I'm calling that PC, equals the pressure in the potential space. And so we can just call that P of theta. And it only depends on theta because we're considering steady flow. Yes.
It's a Peckley number assumption as well, isn't it?
Yes, there is. Yeah. It's about 70. So it is on large side, that Peckley number, but we thought it was okay. I can't remember the calculation, but yeah. That's true. Yeah, and similarly with concentration, they're equal to each other and they're just a function of theta because they're independent. Obviously, we're ignoring variations in the third dimension. Radial we've solved for, and time, we're assuming it's independent of time. So we've eliminated down to one variable, which means that you end up with ordinary differential equations for these quantities. So P is pressure, Q here is volume flow, so that the amount of water going through the choroid, that's volume per unit time. And then concentration of either albumin or drug, and J is the flux of either the albumin or the drug through the whole layer. And this is so these equations. Mu I've mentioned is the shear viscosity, this is geometrical factor, Kc permeability of the choroid, height of the choroid, and Kp here is 1/12, which will be quite familiar to those of you who've done this kind of modelling. Kp, Hp to the m is 3 in Stokes throw. And then the rate of change of the volume flow is a geometrical factor, Hc, height of choroid. Qc is this term that represents the water coming out of the choroidal vessels. Qr is the prescribed flow across the retinal pigment epithelium. Qs is the flow across the sclera. So these two, Qc and Qs, have formulae in terms of other variables, well, P particularly, and Cx. Then rate of change of Cx is given by that times the volume flow, so the volume flow component minus the flux component Jx, divided by geometrical factor. These are the diffusion coefficients, and if you non-dimensionalize, that's where you get Peckley numbers in these two terms here. Okay, so dxc is the diffusion of either albumin or drug in the choroid times the height of the choroid, and diffusion in the potential space, which might be different, but we just put it as the same actually in the model because so many parameter values. It's difficult to know. Okay, so these two are going to be the same as each other times the height of potential space. And then we have rate of change of flux is a geometrical factor times height of the choroid times by the amount of the solute coming out of the vessels minus the amount of solute going across the sclera. And as you can probably understand, these equations are very, very, very much simpler to solve than the full three-dimensional problem or whatever. So it's really worth doing this approximation, and because the factor is 50, it's a pretty accurate approximation as well. And then we have boundary conditions, which I sort of alluded to before. So symmetry conditions at theta equals 0, and then this pressure condition that I've already described, and a flux condition at theta equals theta 0, which is the well-mixed condition. Okay, so results. This is quite a involved slide, so it might take me a little while to explain. This is just physiological conditions, and then examining the pressure and volume and so on and flows. Okay, this I'm sorry, I can't make MATLAB produce this in red. It should be in red, this curve. So this is just the height of the choroid. So it's on this axis that you should be looking here. So the choroid, it's prescribed height, but it turns out the choroid is a lot thicker at the back of the eye, probably because that's where you see most detail. So the choroid is thick at the back and then goes to a much thinner value at the front. And this is in non-dimensionalized units. And then the height of the potential space, we just prescribe it for this value. So I mentioned about the springs, but we prescribe it in physiological conditions, and we use the results of this then to work out the natural lengths of the springs. And then if we change the pressure, we use those natural lengths to calculate the actual lengths of the springs under different conditions. So in this case, it's just, this is at physiological conditions. The height of the potential layer is just 1 in non-dimensional units. And then we have the pressures. So the pressure so I've done 2 cases. I've done no potential space. As I said, it's a bit controversial, its existence anyway. So no potential space in dashed And then with the potential space, you can see it makes a little bit of difference. And the pressure is highest. So the inlet, confusingly, the inlet's on the right because it's at theta equals theta naught. So the pressure is highest at the inlet. And then, of course, because it's driving a flow, the pressure decreases through the choroid down to the back of the eye. Okay. Second graph, this shows the flows. So the flow is, well, in the case with no potential space, this is just this dashed line here is just the flow. So you can see it's greatest at the inlet, and then gradually the flow leaves, it seeps across the sclera, so the flow goes down rapidly through towards the back, where it's very tiny. And then the if we add the potential space, we have then a smaller flow through the choroidal tissue, so it's shown in the red here, and then but then also a flow through the potential space because it's going through a gap, very low resistance gap. So despite being very tiny, the flow is slightly greater in the potential space, and the total flow overall is greater than that without the potential space, as expected. This shows albumin concentration. So at the top here is albumin concentration in the blood vessels, which is a fixed constant, so quite high concentration. The concentration we assume is 0 in the orbit, so that outside the eye. And then we have the actual concentrations with and without the potential space, which are very similar and quite low, which is what's measured as well. And finally, we have the flows of water in the different compartments. So positive flow indicates flow out, flow into the choroidal space. So for example, the RPE flow, as shown in purple at the top, is constant prescribed, as I said. So that is just shown here as positive number because it's coming into the choroidal space. Also, the arteries are at a high pressure, so flow is going out of the arteries and into the tissue. So that is positive as well, and it's shown by the red curves here, almost the same for with and without the potential space. And then from the veins, they are at a lower pressure, and we have flow actually going into the veins, shown here with and without the potential space. And then the flow across the sclera, again, it's going out, it's going across the sclera with and without potential space. There are 37, I think I counted, parameters in this problem. And some of them, many of them we know pretty accurately, but some of them we don't. And so we've done some, this is quite preliminary, but we've done some sensitivity analysis to try and understand the effects, particularly of those parameters that we don't know very accurately because they haven't been measured and are very difficult to measure. So this is, it shows, so for each of the parameters that we have some suspicions about the value, they are listed here. We draw a graph of, for each quantity that is useful to measure, we draw a different graph. And then, so say if we take this permeability of this potential space, that is very sensitive to this one, the choroidal permeability, and then also, not surprisingly, the elasticity of the potential layer, for example. And this sensitivity is the variance of this quantity as you vary each parameter separately. That's what's shown by the solid curves. And then the dash curve somehow accounts, and I don't fully understand this, but somehow accounts for varying other parameters as well. So the dashed lines are always slightly greater than the solid lines. The solid lines, yeah, all sum up to 1 in each case. And so it's the fraction of the variability that is accounted for by changes in that particular parameter value. And so this is work in progress, but this is the way that we're going to cope with the fact that we don't know some of the parameter values very accurately, and we'll look for that will enable us to home in on parameter values that we need to look at. And as I say, that's work in progress. And Jessica Corshaw did all of that analysis. Okay, so that was what I was showing you before, was just for one pressure. But now what happens, the question is, well, if we raise intraocular pressure, because this is something that's actually known about in the literature from those results that I showed you early on in this talk. So here's the same points that I showed you before. How do our results match up to that? So we've considered here, so if I go on the right, we've considered 4 cases. And we've either, so that vessels here indicates we switched off, and when it's plus, it's what I've described to you, but when it's minus, we've switched off the flows coming out of the vessels of both water and albumin. And so the red, so green and black curves show you with no effects of the vessels, and it shows you actually the importance of the vessels in this model. And then we've also actually made the collapsible, the potential space very small in the cases with the minus here. So plus is what I showed you before, the black and blue curves, but the minus here is with the effects of the potential space removed. And so you can see that both having the potential space and also having the effect of the vessels, both of these are really important in the model because you get significant differences. I should mention, I didn't mention at the time, Darrell Overby's original model was with just the potential space. So he had all the flow going through this potential space, and whereas Masha had done the model of just the flow going through the choroid, so that the more recent work's been to unite these two and see what effect that's had. Okay, yeah, so both these factors have a significant effect, and the blue line is with both of the factors included. And if you then translate it onto this curve, this graph, you can see, and we haven't fixed this, honestly, you can see that it matches quite closely with the experimental results, which is nice. So far I haven't done anything with the drug, but if you add a drug into the calculations, you get a profile something like this. And what we find is that at the inlet, the drug concentration is whatever it is. So this is scaled drug concentration, is 1 at the inlet. And then the drug it flows into the vessels along the way and flows across both into vessels and also across the sclera. And so the drug concentration goes down from the inlet towards the back of the eye here. And we have fixed the values in this case. So we've played around, Masha mostly did this, actually played around with the parameter values to get one that didn't drop too much, because for many parameter values it drops by 4 or even 6 orders of magnitude, which is obviously not good for us because we're trying to get the drug to the back of the eye. So we want there to be a significant concentration at the back of the eye. So this is a very optimistic view really here, but it's very much work in progress, and we need to do a bit more on this. Anyway, what's interesting is to see the destinations of the drug. So in yellow, gold, is shown here the drug that goes across the sclera. So that's wasted drug, that's not going to where it's needed. The drug that does go to where it's needed is that goes into the vessels here. So this is good drug, if you like. It goes into the veins and the arteries, which stops the growth of those in macular degeneration, for example. We're doing for time. Okay. So what I've demonstrated in this is that we've got a plausible mechanism. It would explain the unconventional flow, and it sort of seems to fit particularly with this pressure insensitivity that I described that was measured by Bill in those experiments ages ago. If we think about, so one application that I didn't mention is to glaucoma. So because glaucoma is often treated by glaucoma is a condition of high pressure in the eye, which causes damage to the optic nerve. But it's caused by poor outflow from the eye, and it's treated by adding a shunt or something that's low resistance to let fluid drain more easily from the eye. And so if we have a better understanding of where the fluid goes on its outflow, it will help us to understand glaucoma maybe a little bit better, because obviously a significant fraction of the fluid is going out via this route, and currently that's not really accounted for or understood. So that's another application. I've mentioned drug delivery as well. That's an application. We're planning to look at different types of drugs. That's really future work, because we only just put the drug in at all. So different types of drugs do different things, and that's a complicated study in itself. So that will be future work. And also of interest is clinically, it's very easy to change vessel diameters, apparently, by giving a patient various drugs. So you can alter the arterial or the venous diameters separately, but that has the effect of altering pressures in the different vessels. And so this is another thing we would like to do, because that will also enable us to look at the effects on the unconventional flow, and thereby perhaps suggest treatments in that way as well. So that was really all I wanted to say on that. Do you want me to stop now, or do you want
a few minutes?
A few minutes. I mean, I talked about this. So I came a few years ago and I talked about this before, but I alluded to this earlier. So if we're thinking about this drug, this is totally different, by the way. So I did this a long time ago. So this is drugs injected into the vitreous chamber. And I mentioned that it was eye rotations that mix the drug in. And so a long time ago, we looked at the effect of eye rotations on where the drug goes. And the interesting thing was that we assumed very small eye rotations. And so to leading order, considering rotational flow, the eye is rotating backwards and forwards, a bit like you're looking left, right repeatedly in our model, just to simplify eye rotations. And at leading order, the drug is not moved at all. It just goes backwards and forwards with the flow. It doesn't do any mixing. And so actually what we showed was that the leading order transport of the drug, actually the interesting transport, is at second order. Order sort of, if the amplitude of those rotations is order epsilon, so order epsilon squared, that you get interesting flow. And then you get these big streamlines, so that's how we'd expect the pattern of drug flow. So if you put a blob of drug over here, for example, it's going to be pulled along to the center line, and then it will go up and down along this axis and then round. And that's been demonstrated in experiments as well. Clinically, the useful thing we can calculate from that is a kind of time scale that it takes the drug to get from where it's injected to the back of the eye, which is useful clinically. Yes, so that's helpful for these intravitreal injections. We can also, from this flow, understand, from the leading order flow, understand the stresses, and that's important for retinal detachments. That's something else I've worked on many years ago. If anybody wants to ask me any questions about that, obviously you're welcome. Another thing I've looked at in the eye is this flow of anterior, of the aqueous humour in the anterior chamber. And I both looked actually at flow through this very narrow gap between the lens and the iris, which is not shown to scale in that picture. But here, this is just the flow within the anterior chamber, which has, as I mentioned already, there's a radially outwards flow, but there's also a flow due to eye rotations, very similar to the flow that I showed you in the vitreous humour. It's the driving mechanism is similar. And then there's also in the anterior chamber a thermal component. So typically you're in a room that's at a lower temperature than body temperature. And there is, as in a room at 20 degrees Celsius, is about a 3-degree temperature drop. So it's slightly warmer at the back of the anterior chamber than it is at the front, about 3 degrees. And that causes the slightly colder and thus heavier, denser fluid at the front is going to drop. And the warmer and less dense fluid at the back is going to rise, and you get an overturning instability. And so I've done some work on that. And actually this thing here, this white thing here, is an intraocular lens used to correct vision as an alternative to wearing glasses. But anyway, the important thing is that you get this flow. And actually these two flows are both of the same order of magnitude. and much larger than the radially outward flow, actually. So these are the predominant flows. Obviously, this one, if you're asleep, if you've got your eyes shut, switches off because your eyelids are heated. So the flow in the anterior chamber is very complex and different at different times of the day as well. That's all I wanted to say. It's just a little, if anybody wants to talk to me about those things. This work has arisen out of a special interest group, which was originally called Fluid Mechanics of the Eye, and it's now expanded to include non-fluid mechanics people as well. So we're now calling it Virtual Eye Consortium. And originally we were funded by the UK Fluids Network, and more recently by the Macular Society, and then the Newton Institute as well. We now have funding from them. And you know, if anybody here or you know of anybody that might be interested in joining that, it's open to everybody. And there are online meetings every so often, but we also are going to have an in-person meeting in Glasgow in January. So if anybody is interested in this kind of research, you're obviously welcome to come to that. Thank you very much for your attention.